Hello, welcome to another edition of the CNA's Obits. My name is Rich. I'm a certified nursing assistant. And let's see what we have on today's agenda. Alright, okay. This here is ubiquitination. What happens is when proteins need to be broken down in the cell, they get ubiquitinized, and then this little machinery within the cell called the proteasome will chew it up and spit out little fragments of it. But there's one thing in this that I kind of want to make a connection with, and you'll see it in a second. This up here, this is the p53 tumor suppressant protein, if you can see it, and that's MDM2. And the MDM2 will try to bind to it and inhibit it and ubiquitinize it, so it's targeted for destruction. But you'll notice that to prevent this ubiquitinization, a phosphate group is added. There's a phosphate group that's added there. I got a new camera here, so it's a little messed up. There's a phosphate group. It's added there, so it's not targeted for ubiquitinization. Now, if you go down here, that is beta-catenin, and beta-catenin goes into something called the beta-catenin destruction complex. And these are several proteins on the outside here. Here's axin, that's adenine polyposis coli, and that one is a pretty serious one because mutations in that can is a direct link to a familial form of cancer. But that's not on the topic right now. So these are scaffolding proteins. So basically, they just hold proteins in place. These two outer ones, the APC, the adenomous polyposis coli, and the axin, they just hold proteins in place. They're scaffold proteins. One of them that they hold in place is glycogen synthase 3 beta, and then on the bottom here in red is casein kinase 1 alpha, and they're both serine threonine kinases, which means they add phosphate groups to serine or threonine. Now, when beta-catenin comes in here, it gets a phosphate group put on first by the casein kinase 1 alpha here, bottom one, and that sets off a reaction where the glycogen synthase 3 beta will put on phosphate groups down here, going downstream towards the amino terminus down here of the beta-catenin. But the beta-catenin destruction complex does not target it for ubiquitinization. It then leaves there and it gets targeted for ubiquitinization by an enzyme called beta-TRCP. But you'll notice the difference between the two here. This one didn't get targeted for ubiquitinization because it was phosphorylated. This one is going to get targeted for ubiquitinization because it is phosphorylated. So the exact opposite thing is happening here. Now, if we go down here, let's see this. This is hypoxia inducible factor 1 alpha, and in there you're going to have two prolines. You're going to have two prolines right there on it. And this enzyme, during normoxic conditions, when there's enough oxygen for the cells to survive under normoxic conditions, an enzyme called propyl hydroxylase will come in and add, see here in blue, that originally isn't there. It adds a hydroxyl group to the fourth carbon. So it adds that there's an oxygen there now, where the original proline does not have that oxygen. it adds that during normoxic conditions within the cell. And that's called 4-hydroxyproline because it's off the 4th carbon. Then tumor suppressant protein, von Hippel-Lindau protein comes in. See, little p, tumor suppressant, von Hippel-Lindau protein comes in, and that targets it for ubiquitination. When both these prolines, here and here, both have a hydroxyl group added to it on this 4th carbon here, the von Hippel-Lindau tumor suppressant protein will come in and add a ubiquitin group onto it so that it's targeted for ubiquitination by the proteasome. But you'll notice the difference between all of them if you could look at it as a general theme or a general untheme. However you want to look at it, is that the p53 protein, tumor suppressant protein, is not ubiquitinized because a phosphate group is added to it. This one, the beta-catenin, a ubiquitin group is added to it and it's targeted for destruction because phosphate groups are added to it. And this one down here, the von Hippel-Lindau tumor suppressant protein adds a ubiquitin group onto it, and there's no phosphate groups added to it. It's actually just a hydroxyl group that's added to the 4th carbon on both of them that brings in the von Hippel-Lindau tumor suppressant protein. And it's hydroxylized by propyl hydroxylase, the enzyme right there, that comes in, does it during normoxic conditions. But what you're seeing is that if you want to look at it as kind of a dogma in a sense, is that the starting point of ubiquitination can be entirely different all throughout the cell. So certain things are made, like p53, beta-catenin, hypoxia-inducible factor 1 alpha are constantly being made. Those are all proteins that are constantly being made, and they're constantly being destroyed. And they're only not being destroyed when necessary, when the cell needs them. Say, for example, with p53, when DNA needs to be repaired, or in hypoxic environment where HIF1 alpha needs to bind with HIF1 beta, form a transcription factor for angiogenesis, so we get more oxygen, more nutrients to the cell in a hypoxic environment. So these things are all constantly being made, and then they're all constantly being destroyed until they're needed. But the point is the point that I really saw here and trying to make a connection here is that the starting point of ubiquitination can be different all throughout the cell. It isn't just adding a phosphate group. It isn't just not having a phosphate group. It isn't just adding hydroxyl groups. It's the starting point can be all different. And that's really all I wanted to say, is get that point across. Rich, see you later.